So as I was saying, my name is Mario Thomas. I'm a Senior Vice President at Ontario Centres of Excellence and Managing Director of the Centre for Commercialization of Research. Thank you for joining us this afternoon and welcome to Discovery. Before we go any further, I'd like to acknowledge the generous support of Bruce Power, the sponsors of this presentation theater. I'd like to start our session this afternoon by introducing Dr. Donald Stuss. Don is the President and Scientific Director of the Ontario Brain Institute and has been a University of Toronto Professor of Medicine and Psychology since 1989. He was founding director of the Rotman Research Institute from 1989 to 2008. His personal research focuses on understanding and treating the cognitive functions and personality changes associated with the frontal lobes as they occur after stroke, in normal elderly, and in those with traumatic brain injury or dementia. Welcome, Don. Before we continue with the panel discussion, which Don will moderate, Don is going to announce a new joint Ontario Brain Institute, Ontario Centres of Excellence initiative. Over to you, Don.
Okay. Thank you, Mario. So let me start this symposium off with on a very positive note. One of the major challenges in commercialization of research, and notably I think neuroscience research, is the lack of sufficient managerial talent with an entrepreneurial spirit. So we've responded to that challenge. The Ontario Brain Institute and the Ontario Centres of Excellence have joined forces to give some of Ontario's top young entrepreneurs the chance to turn their discoveries into viable neuroscience-based businesses. Through the OBI OCE Entrepreneurial Fellowship Program, postgraduate and early-stage entrepreneurs will receive $50,000 for 1 year to support the commercialization of their discoveries to help diagnose, treat, or cure brain conditions. So this program aims to support the growth of the Ontario Neurotechnology Cluster and to mobilize the Ontario-based research community and industry in a mentorship advisory capacity. Applications can be accessed on our website. It's www.braininstitute.ca, or you can get the information as well on the OCE website. Application deadline is tight, because we've been moving very quickly on this, is May 28th of this year, and we expect that candidates will begin the programs on the 25th of June of this year, 2012. So that's our good news. We're very pleased to announce that, and very pleased to show how a cooperative spirit can come up with innovative approaches. And now I'm delighted to welcome you to the panel discussion, Neuroscience, Challenges and Opportunities in the Commercialization of Research. Canada's got world-class neuroscience talent. Ontario has world-class neuroscience talent. The province of Ontario recognized the potential and in 2010 established the Ontario Brain Institute to create an environment that would maximize this talent to create significant commercial and health impact. To achieve this, OBI created a research environment that was patient-centric and interdisciplinary and broke down barriers between clinicians and researchers, between institutions, between research and industry, and even between diseases. We did this by bringing everyone together early in the development of initiatives. But we quickly recognize that many barriers continue to exist. How do you deal with the complexity of diseases and how they manifest and progress, including comorbidities and genetic variations, and the desire for personalized medicine? For many of the brain disorders, are there adequate models of the disease to do the foundational research required? The pipeline from model to product has holes and blockades. One clearly is finances, but there are others: ethics, regulatory bodies, relative risk. And here's a hard one. How do you measure success? In science, health impact, transformation, and commercialization. So you will see that our panelists were chosen wisely as they debate these types of issues. So we have two main objectives in today's symposium. One is better understanding of the challenges related to commercialization of neuroscience research and technologies. And the second is hopefully to give you increased knowledge on how to overcome these barriers and look at how we can adapt best practices from others through collaboration. Now, before we start, I'd like to share with you the structure of how we're going to work this afternoon. We're going to first start with giving you an introduction of each of the panelists summarized in a slide. When I introduce each individual, they will give a high-level resume of their background and relevance to this session. I hope you recognize, as I did, that this panel represents excellence in achievement and brings different experiences to the question, providing a variety of lenses through which to view the challenges. The core of the session will focus on 5 challenges in commercializing neuroscience research, some of which I touched on above. For each challenge, one of the panelists will start the discussion by setting out what that issue presents, why that issue presents a hurdle for commercialization, following by, hopefully spirited, dialogue among the panel. At the end of this panel discussion, we will then open the floor for an open session, including questions and comments from you, the members of the audience. And now I'd like to bring the panel up, please. So our first panelist is Dr. Max Cynader, Director of the Brain Research Center at the University of British Columbia, somebody whom I've always admired for the energy. So welcome, Max. And we have the first slide and some background introduction. So Max, I'm going to turn it to you, and you can just stay seated and talk right.
Stay seated, okay. All right. Hello, everybody. I'm Max Cynader. I'm a neuroscientist. I'm director of the Brain Research Center at UBC and Vancouver Coastal Health. And, you know, I feel like I'm at an Alcoholics Anonymous meeting because I'm going to admit it. I'm a serial entrepreneur. I've actually, in addition to doing fundamental research all my life, I've started 2 companies, and I was telling Michael and Kelly earlier on that I'm thinking of starting a 3rd, actually. I just wanted to introduce a few points that are relevant to commercialization from the perspective of a startup, someone who's starting a company, basically a scientific entrepreneur. You cannot do without strong science and intellectual property. If you don't have that, you're not going anywhere. But you must, in addition, have you must find partners. You must find people who will complement your strengths. You must create situations in which there really can be win-win. You must put yourself constantly in the shoes of the people who are giving you their hard-earned money, who may be interested in buying your products if you ever have them, and who want you to succeed by being your partners. You've got to put yourself in their shoes, and you've got to stay there. I've got an additional point, choosing your market indication carefully. You know, we all want to achieve home runs. So when I started my first company, it was grandiose. It was Alzheimer's, cancer, and glaucoma. And of course, we succeeded at none of those. But we succeeded anyway, because we eventually learned that if you choose your indication very carefully, you have a much better chance to succeed than you do if you simply go for the biggest hit possible. The other thing we learned is that you have to give yourself more than one way to succeed. If you can think of a way to take your technology, your idea, and move it from a single compound, a single indication, a single strategy, into one that is a platform, that will be enormously to your benefit. And finally, I want to remind you of the 5 M's, which are essential for a startup. They are money, 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 management, and money. And I think that's all I'm going to say. You don't have to show the second slide. I'll save that.
Okay, thank you. No location?
Pardon me?
No location, just money, money.
Money, and
management and money.
And those are the 5 M's, yeah.
Thank you, Max. And next to Max is Dr. Michael Fehlings. He's professor of neurosurgery, University of Toronto, and head of the Krembil Neuroscience Center. Thanks for being with us today, Michael, and tell us a bit about your background.
Well, how do I follow that act, right? Reminds me of the movie, Show Me the Money. So I guess I'm here as a neurosurgeon who treats patients with neurological disease and as a clinician scientist, and I guess I'm kind of bridging a few different areas of activity as an academic neurosurgeon with a focus on neurotrauma and spinal disorders. I come kind of representing a number of networks, both at the University of Toronto Neuroscience program, as Associate Scientific Director of the NeuroDevNet Network Center of Excellence, and as the lead stem cell scientist in one of the OBI-funded initiatives, CPNet. I've led a number of clinical trials, and actually the Chief Scientific Officer of a clinical trials network called SpineNet, and I'm the principal investigator in the Christopher and Dana Reeve Foundation, North American Clinical Trials Network, and we currently have a number of strategies that are in the pipeline. And my home base is at the Toronto Western Research Institute. I guess I have a second slide.
Go ahead.
I was asked to provide 2 slides.
So. Okay. There we go.
There we go. Okay. So these are just some of the areas of active clinical research. So I was involved in bringing a recombinant Rho protein antagonist from molecule to man, and we're currently just finished phase 1/2A clinical trials and are now moving forward along that pipeline of discovery. I've actually have a number of patents in the area of spinal implants, and they actually make money. And I'm involved in the development of an osteobiologic called P15. So those are some examples of commercial translation of research, and then I'm involved in preclinical translational discoveries with a kind of a major focus around developing unique models of CNS injury and disease, and then trying to test the efficacy of these novel therapeutics. So I have some involvement both at the preclinical level and then the clinical translational level.
Thank you, Michael. And you can see the strength that we have in our panel. I thought you did pretty well there, following Max. That was good.
It's a hard act to follow, I have to admit.
So now, money, money, money. I'm going to turn to Kelly, co-founder and managing director of Genesis Capital. Thank you for joining us, Kelly. Give us some background, please.
Yeah, thank you very much for the invite, Don. My name's Kelly Holman. I'm a general partner of a Toronto-based life sciences group called Genesis Capital. Over the last 10 years, we've raised about $225 million, invested across 26 companies, and of that 225, we put 125 to work. I'm a board member of 2 companies presently, one in the area of neurotechnology, functional neuromodulation. When I've looked back and in our own history as to how many times, I haven't thought about allocating to neurotechnology. We tend to allocate it around different risk profiles, different stages of development and the like, but I highlight 3 that are in our current portfolio. Functional neuromod, NorX, which is a functional neuromodulation, some IP sourced here from the Toronto Western Hospital. NorX is IP sourced out of Northwestern in Chicago, and Noraxon, which is a company based here in Toronto with some operations in Boston. Of the 11 companies, 3 are in neuroscience, these 3, and over the 26, 5 of our companies have been in the neuroscience, neurotechnology area. It's, as we will discuss, it remains to be a tantalizing area to invest in, yet with an enormous amount of risks associated with developing it to a commercial success, profitable enterprise. Thank you.
our points of discussion, how important to have all of these different perspectives. And finally in our panel is Toddvino. Todd is the Director of Business Development, New Growth Opportunities at GlaxoSmithKline. And I'm also very pleased to indicate that Todd is the Chair of the Industry Advisory Council for the Ontario Brain Institute, helping us deal with some of the issues of commercialization. So, Todd, please.
Thank you, Don. As Don said, I work for GlaxoSmithKline. I've been in industry for about 20 years now, 20 plus years really. I kind of want to stop counting, but 20 plus years. I currently am in business development, but over the years I've done a number of different functional roles, from medical affairs, regulatory affairs, and pharmaceutical development, leading teams in each of those areas. So my background is fairly broad in terms of the different types of technologies and areas that I've touched. And including in therapy areas. One of the therapy areas I've worked in and more recently worked in is neurosciences, which is how I got connected with the OBI and with Don, and my role within the OBI of the Industry Advisory Council, and now as a member of Board of Directors as well. So that's my background. Just to give some sense of what's going on in industry. Next slide. So things in big pharma are changing. I think there's an evolving pharma model, and the needs of the market and the demands of things are forcing some change, and I think it's relevant to this, so I'll talk about a few of them. First of all, as any business, we have to keep generating our top line results, and as patents end and we see some products come off patent, we have to have a backfill of products to go behind that. And that's not always easy. In today's world, we lose some fairly large products in our portfolio, and they're backfilled with smaller products. So we need more of those to backfill those big products. And so as those demands and those pressures on the commercial world are happening, it really affects how we look at our pipeline. And the pipeline isn't just necessarily comes from within our company, but we also look outside our company to find opportunities to expand and grow our businesses. It's really important to note that payer perspectives are also playing a much larger role than they used to. It's no longer just about getting products approved from a safety and efficacy point of view through regulators. You really have to look at the reimbursement environment, and is something going to be paid for? Everybody's heard these days the constraints on the healthcare system. Everybody understands that we're on a path in our spending model that isn't going to be sustainable if we keep the same trajectory. So everybody's looking for ways to save money in the system. And so drugs have to reach or products in general, health technologies in general, need to achieve a fairly significant hurdle rate for getting reimbursement these days. You have to take that into account. At the same time all of that's happening, development costs are increasing. They estimate these days it's nearly $1 billion to get a product to market, and it's hard for a small company to achieve that on their own, so we need partnerships to happen. So big pharma has a pipeline need. Small companies can't afford to do things on their own. It's ripe for partnership opportunities, and I think that's important for people to understand at both sides. And in reality, when you're getting pressures on the top line of a large organization, where we go is we look at how can we save money and expenses as well. So we have to focus our R&D money in places where we can make it most effective. And some of these big challenges, the universal questions in disease areas that we all have, we can't achieve those as a single company. No one can afford to do that anymore. So we are looking for those areas where we can partner with other companies and governments and the public-private partnership type opportunities where there's ways that we can find these things out in a pre-competitive space. That's also something we've seen a lot in the trend. So lots of opportunity to look for those partnerships, and big pharma is open to these types of partnerships probably more now than they ever have been. So I think that covers most of what I really wanted to talk about. The other points we'll get to in the other discussions.
So thank you all, and I hope you get the sense again of two issues here. One is the fact of the challenges that do exist, but also I think you hear an underlying current of optimism. And it's why the reason that we invited these individuals to be on our panel. So as a reminder, just to tell you a bit how we're organizing this, is that we have 5 topics that we've kind of have selected as we think are representative and major, and obviously you can ask some later. We're going to break these down over the next hour into kind of about 12-minute segments. I'm asking one of the panelists to kind of lead off the area, and then the other panelists will join in. We're going to leave questions and answer till after we go through these, and then we'll open it up for general discussion. So the first thing you've heard about, we've talked a bit about, is the whole issue of a model disease. Like, how does one develop when you may not be sure that the model you're working on may be the right one, or maybe even allow you to develop the kind of pipeline that you need? So I've asked Michael to kind of pitch in to start and lead out the panel off on this, and then it'll be open. So, Mike?
Right. So the development of accurate models of disease is absolutely critical, and it's I think one quickly needs to get into specifics when one is talking about CNS models. But one of the challenges with any type of a model is that it needs to reflect the heterogeneity in the patient population, and that whether the patients are young or old, the genetic variability, the disease comorbidities, et cetera, that are involved with this. And so it becomes quite challenging to model this accurately. And then, do we rely solely on in vitro testing? To what extent is in vivo testing required? Do we go with small animal models? Does everything need to be replicated in large animal models? And we have to look at the invasiveness of the types of therapies that are involved. In certain circumstances, such as traumatic brain injury or spinal cord injury, we know the cause of it, so we understand the cause. In other areas, schizophrenia, autism, we're only now starting to unlock the root causes, and so this becomes important. But one thing that I think is really critical to this is the linkage between the clinical cohorts as well as the preclinical basic science. And so, if you will, kind of a model of both forward and reverse translation. So being able to, for example, identify genetic predispositions to certain diseases, perhaps to develop animal models of these, and then to validate these again in terms of clinical trials. So I think that we have a number of models in the central nervous system, but it's an area I think that requires a great deal of attention, and I think we need to have a greater linkage between these clinical studies and then the preclinical studies, and going back and forward.
Anybody else on the panel have comments?
Well, I would add that for us, modeling is extremely important, because having confidence in assets as they move through development, if we don't have a model that we're confident in, then we're reluctant to invest. We're reluctant to really put more money into it and say, well, we know that this is going to have the benefit that we think we'll see in humans. So you can go not just from moving into clinical, from preclinical to clinical studies, but also in terms of relative benefit versus other assets as well. So there's a number of aspects about animal modeling that help us to determine whether or not we want to go forward fast, or whether we want to go forward at all. If you don't have that as a foundation, it's a huge hurdle to want to put the money behind a clinical trial that's going to give you that information. So modeling is extremely important to us in terms of the pharma industry.
Hey, Max, you.
Yeah, so I'd just like to echo Todd's comments. One of the points on his slides, on his slide that I looked at before, was the words "fail early," and I really, if you're going to fail early, because the entire process of commercialization becomes much more expensive as you march through the steps. The preclinical part, the animal models part, is dirt cheap compared to the overall costs of the entire effort. So it really, from the perspective of a founder, a company founder, it really behooves you to do the best possible job that you can at the preclinical stage. It's cheap. It gives your partners confidence in what you're doing. We have now seen in the last few years situations in which there are okay preclinical efforts. They're good enough to lead the startup to an IND, a phase 1, maybe even a phase 2. And that's the point at which it fails, when the startup has realistically, very few startups have the capability of running a phase 3 in any disease. So you've got to have a partner, certainly by phase 3 and probably by phase 2. So if you haven't done your homework at the earliest stage, if you haven't respected the variability, the complexity of the disease that you're trying to tackle, you're going to get hit. The penalty may not arise immediately, but it will arise when you try to go for that second or third round of funding or that major pharmaceutical partnership that is necessary to get your product to that next stage of clinical trials.
Kelly?
I would just say, I mean, relative to other areas of life sciences or other areas of diseases, neuroscience, I think, does suffer from having, I mean, we can all argue how predictive in vivo testing will translate into humans. It certainly is a basis, certainly has to happen before a regulator in the Western world likely will give you permission to do some human testing. But I think in neuroscience, it certainly suffers more than other areas on how much confidence you can gain from that data. It's nothing we're going to solve today. I think the comment though is bang on. I think the more experience you do get it, can you be better informed in coming up with those models with some of the human experience and feed that back into development.
So I have a question I was going to ask Michael that when you started off, you said two things I found interesting. One was that it's very difficult to find models of certain diseases. So trauma we know, but depression, I mean, we have models of that, but and there are animal models, but are they the same? What do they do? And the second one, which I found very fascinating, was this back and forth, that we need both the reverse and forward. And I'd like to open both of those issues up to the panel, because I thought those are potential answers or lead us down pathways.
So I would argue that it's not just a simple linear transition from animal models through phase 1, 2, and phase 3 studies. There is no perfect animal model of any human disease that I know of, and even, let's say, in an ideal circumstance such as traumatic spinal cord injury, which is what I work on, where we really have excellent models of the disease pathology, they still don't really mimic what occurs in clinical cohorts. I think that having disease models that reflect the heterogeneity of the pathology that we see in man, more accurately defining the pathology that we see in man, not always trying to hit the home run, having one disease model trying to hit all of the disease entities. So stroke is not stroke. Stroke in the young is not the same as stroke in the old. The middle cerebral artery occlusion is not the same as a brainstem infarct. It's just not the same. And so, having, realizing what you're looking at and probably being more targeted in the therapeutic approaches, and then also respecting the genetic variability. And we've seen errors that have occurred where, for example, we've failed to respect important sex differences in the pharmacology of drugs, et cetera, et cetera. And so I think one of the points here to get around that challenge is to really reflect on what the phase 1 studies are telling us, to perhaps pay more attention to the phase 1 studies than simply looking only at safety and feasibility. Trying to look at signal, trying to understand the heterogeneity of the cohorts, going back to the animal models, going back and forward, and paying a lot of attention to the smaller phase clinical trials, and then trying to parse out which disease models are going to more accurately reflect what's going forward, and not to jump the gun and start going into a phase 3 trial before you fully understand what it is that you want to be looking at, what is the patient population, what is the outcome measure, et cetera. You can learn a lot of these in carefully targeted preclinical studies, and then looking at these in limited patient cohorts. This is what I was referring to in terms of the forward and the backward translation.
Anybody else have a comment on this? Because from what I understand, that's not commonly done. Once the pipeline starts, it goes and then it fails or advances.
We need to look at alternative models. So, and stroke has been a perfect example of where that classical pipeline has not worked.
So I'll comment on the fact that we're talking here a lot about preclinical models, and I think that's fair, but there's also, when you bring up depression, one of the issues is actual clinical endpoints. And clinical endpoints in studies, if we don't have good clinical endpoints that we can use to measure, good tools, whether they're animal models or tools that we use, that creates problems as well. And so we need it not only in the animal side, but especially in the softer endpoint type of point, like depression, for example, we really do need really good tools.
So that actually moves us. There are two topics that came forward that I want to talk about that actually hit upon our points. One is variability, and I think that behooves us really to come back to that. But I'm going to follow through, Todd, just with what you mentioned, is on the measurement of endpoints, because that is one of our topic areas. And Max, I'm going to ask you, because as a pioneer in many areas of brain research, to give us some of your experiences and background about how do you come up with adequate endpoints? What is what you're trying to measure to show efficacy?
Well, I think part of this has to do with the state of the science, and I think I'll just follow up with what starting from Todd's points. So, for instance, we're about to have in the whole area of psychiatric disease, we're about to have a new psychiatry Bible called DSM-5. And it's going to, again, completely change the way in which patients are categorized for diseases. And what it does is it reflects what we're learning about diseases that used to have names like schizophrenia and depression, which are, again, the kinds of home runs. So depression, for instance, has been, considered to be sort of like it's like a Chinese meal. You've got a whole list of symptoms on page 1, and if you've got any 2 of those, and then there's a whole list of symptoms on page 2, if you've got a whole list of those, if you've got any 2 of those, so 2 from page 1, 2 from page 2, and you're depressed. But you can have 2 different patients who have no symptoms in common at all, and yet they're both classified as depressed. Now, what makes you think that any pharmaceutical that you've got, even if it's really good, is going to be equally effective in those 2 patients? It can't be. So your measurement of the endpoints in this one example is obviously, we need a different set of endpoints. People are now talking about looking at each individual line on that Chinese menu and taking each one of them as an endpoint. That's a scenario, but that may not be enough to give you clinical efficacy. But at least it gets you some of the way. So I think we need better endpoints at every level in the process. Schizophrenia, it's a similar kind of story. Why don't I stop there?
No, that's good. But at every, because you're saying something else that's important, at
Yeah.
every level of the process,
yeah.
which goes back a bit to some of the other conversation on the back and forward, that if you get to a point and all of a sudden you see that there are different subgroupings, you may come back and then change the formula based upon this group with this model of depression, these 2 points, and then start again, and another group starts again.
Yeah.
The goal is to get to an objective response metric, because I think right now what you've got in many areas is it's driven by the patient, and which leads to high placebo. And so, I mean, again, neuroscience, a lot of different diseases and a lot of different things, but certainly in depression, pain, you're asking the patient, and the patient's telling you how they're feeling, and that's why you do see the high placebo. You're not sure. I mean, ultimately, you do. It's I think the goal here is to find the objective response or a functional biological. I don't know if you want to call it a marker or an event that not only shows it works mechanistically, but and then you can tie it to some of the feedback from the patient. And I think that's where a lot of the teasing needs to happen. And
rather than perhaps we've been giving you a little too much gloom and doom, but I should also tell you that as a neuroscientist, I've been doing neuroscience research for almost 50 years now, and I can tell you that the field has never been moving as rapidly as it is today. We are at an extraordinarily exciting point in the development of neuroscience. In particular, I'll just mention imaging technology as a way to get to accurate endpoints. So one of the reasons we've actually been pretty successful at bringing drugs to market in the area of multiple sclerosis is because we have objective, rigorous, well-defined measures of the demyelination that occurs in multiple sclerosis. If you think about it, if you've got foci of demyelination in different parts of your white matter, you're going to have totally different symptoms. Your ability to come up with accurate assessment of how debilitated someone is will actually be rather weak if you just ask him, how do you feel? Or if you assess, if you assess his vision and there's no plaques on the back of the brain, well, you won't get much. Same with motor. But if you look at the entire brain and look for objective measures, the same kind of thing is happening in the area of multiple sclerosis. We now have the development of the Pittsburgh compound and other compounds to measure amyloid load in Alzheimer's disease. And this is, by the way, an opportunity for you budding entrepreneurs. If you can think of ways to give us better measurement systems, better ways to measure endpoints, whether your endpoint measurement is imaging, well-curated cognitive test, a epigenetic mark, whatever you can think of is okay. But that reduces the variability in our measures. And in the end, that's what we're trying to get to. If you can have a much more precisely defined set of endpoints, that makes the clinical trial that much cheaper. It's just, the variability, that sigma measure is direct. I think it's directly or the square related in a square manner to the cost of the clinical trial. So the better we can be at measuring those endpoints using these kinds of ideas, the better we will be in the whole process.
But also in that, it's coming back to where do you have confidence in your investments? Because if not only does it cost more when the variability is higher, you're not sure if you're really going to see the difference you think you're going to see because the variability is so high. So it's a vicious circle in terms of how you can make investments in places. And if you look at, if you can only choose you have a certain pool of money, you can only choose so many different areas to research. You're going to research in those areas where you have the most scientific confidence. So your models and your endpoints and all of those things build into your scientific confidence. And if you don't know, if you don't have enough confidence in your opportunity, even if you don't have a compound, or is where you're going to look for compounds, it's where you're going to be able to measure your success with that compound. It all trickles all the way back to the very beginning. And so all of these things weigh on the whole issue of where do we want to put investment and how is that going to show advances in science and advances for patients who need it.
Goes back, Michael, to what you were saying.
Yeah, so I mean, I think it represents one of the biggest opportunities and challenges in neuroscience. So MS is really one of the prototypical examples, but unfortunately, imaging plaques doesn't apply to most other areas. It is a great example of where a rigorous surrogate endpoint has allowed the more rapid translation of therapies. I think it represents an opportunity for us to link the animal models more closely with the clinical model. So, are there biomarkers either at the protein level or at the imaging level that we can identify from animal models that can then be tested and replicated in clinical cohorts? It's going to cut down the the size of the clinical trials. It'll allow us to fail earlier as well, which I think is also will also be very important. It's very difficult, having said that we should be trying to detect signal at the phase 1 level, it's actually very difficult to detect signal at the phase 1 level unless you have these kinds of surrogate outcomes. So I think it represents an area of opportunity, and I think it is something that we haven't spent enough time trying to develop. And again, I think this points to the critical importance of trying to link the animal models with the clinical cohorts.
So one of the things that I'm hearing as a take-home message, I think one of the things is the fact is that commercialization can occur in the endpoint itself, and developing the endpoint.
Absolutely.
Do it really well and come up with something that is hard and concrete. In fact, would have enormous payoff in related to whatever disorder one is doing, and that one may look at different types of these hard concrete measures. I think that was an important lesson that I've heard. I think the second is that be careful about going down the line. I like this idea about coming back, because if you do find that there is a group and it doesn't work, you can still come back and then start again, potentially with a different endpoint, depending upon what you're looking at. So defining exactly what you want to get at. And I think the third I want is, you've been in neuroscience for 50 years. I want to take whatever you're taking. Wow, that's good. That's my lesson for today. I thought that was. So
Actually 48.
48. So there's a lot of coherence that's happening here, and I want to tie something else that comes very close back to, and it's really our third topic, and I'm going to give a little bit of my own research anecdote that leads into it. Is I was doing a study in patients who had white matter disease, and the normal publications for years have been, the greater the white matter pathology in the brain, the more these functions deteriorate, a specific set of functions deteriorate. And so I do a study in white matter pathology in which the patients are drawn from a different population than normal. We drew them from a stroke clinic rather than from a memory clinic. And I find no change related to the amount of pathology in the brain in comparison to function. It goes against everything that had been published. And one of the reasons was, as we looked at it closely, was there was a subgroup of people who had the most white matter in the brain who performed perfectly normally. I take them out of this analysis and I get exactly the findings. So it's this idea of a disease is not a disease. The white matter pathology ends up being a description. And how many of those? So I want to open it up now to the panel about this whole issue of noise and variability in the system. But I'm also, what you have said to date is suggesting that you have to look at noise and variability at many different levels. So I'm going to ask, actually, Michael, you started that a little bit, so why don't you push that a little bit more on this noise and variability?
Well, it's a critical point, and in the setting of, if we look at say a typical animal model, if I look at my own area in spinal cord injury, we typically will study this in a given strain of mouse or rat. We control for the sex, we control for the type of injury, we give a very precise, calibrated type of an injury, we use very set time points. Now we try to translate this into a clinical trial. We're looking at young and older individuals with varying types of traumatic injury, with varying genetic backgrounds, and so on and so forth. And there's a tremendous heterogeneity. And this brings in one of the areas of complexity. So one of the areas that I think we can do a better job of in the preclinical models is to actually embrace that heterogeneity in terms of the models, and to actually try to use bioinformatics approaches to actually understand that heterogeneity and to try to actually develop a systems-based approach toward doing this. And there actually have been efforts to try to model this in the setting of neurotrauma, so actually to embrace that complexity. And in the setting of the clinical cohorts, I think that the more we understand that heterogeneity and the basis of that heterogeneity, the better we're going to be able to target the different therapeutics approaches.
Yeah, so I would basically agree with what Michael says. And I think there's a theme that is emerging here, and Todd, and Kelly have pointed out, some aspects of the same idea. Basically, if we can be more accurate in our assessment, be it of endpoint or the point I'm going to come to is in our inputs at the beginning, then the entire clinical trial process gets cheaper, gives the large companies much more confidence in what is going on. And I would make the argument that neuroscience is not yet as good as other areas of biology yet in terms of its ability to stratify patients. What we're learning is that many diseases that have the same name are actually extremely heterogeneous. Cancer is a great example. By now, in our environment in Vancouver, many patients' tumors are being sequenced. And we're discovering that there's a tremendous amount of heterogeneity within an individual patient's tumor, within patients who have the same, they have the same so-called disease, but things are actually very different. We've learned in the area of autism, at least in part because of work that was done right here in Toronto, that there are probably 100 different genes that can go wrong and lead to the same so-called disease, autism. Well, I would bet you that you're not going to have the same treatment for a disease that is caused by a malfunction in one gene as a disease that's caused by a malfunction in another gene. So you might as well really start to take note of what that patient actually has wrong with them. My day job, as Don mentioned, is director of the Brain Research Center at UBC, and we're now opening up the new Javad Moafagian Center for Brain Health, which is really a translational facility. The idea is to bring together the major clinics for neurological, psychiatric diseases, bring it together with the healthy aging program and rehabilitation medicine and geriatric medicine as well. But the dream is to treat every patient as a research subject. So we're developing, for instance, a neurodegeneration chip. It's got 250 genes on it. We're doing exome sequencing of every gene that has ever been linked with neurodegeneration in any way, shape, or form. You can do it now for $120. We're working on a synapse chip, which will take the 800 genes of the synapse and put those on another chip and put those together with patients. The idea is to really stratify the patient populations at the outcomes, at the outset, see what kinds of outcomes this leads to, reduce the variability in subsequent clinical trials by knowing much more about our individual patients. And I think this is the way things are going not only in neuroscience, but we're actually behind in neuroscience. So I think that the opportunity to do this better lies before us.
Comments?
No, I think if one of the ideas that I think, Max, what you're talking about is better controlling the input, so the inclusion criteria, if you will, and making it strict. I think and it'd be interesting to get the big pharma approach, because I think big pharma kind of likes the which is if I can
understand.
But from an investor, I think as long as there's a big enough market for someone like Todd may find as niche-y, if using that strict inclusion criteria, we can advance something to start answering the questions. If there may be broader applicability, fair game, that's even better for us. But if there's a real, if we can run the experiment, get some results, and see a commercial opportunity for that patient population, I will turn it to you, because I've always found this dissonance within big pharma, with all due respect, which is they embrace the pharmacogenetic, but it argues against the empirical model of pharma.
Yeah, absolutely.
So it comes back to personalized medicine in many ways, which is around, do we really understand the makeup of the person that we're treating, and what do we know about that makeup and how they will respond to a therapy or the nature of their disease because of their personal makeup. So going back to your comment, in our world, when we design a clinical trial, we try to de-risk the trial. I mean, in de-risking the trial, you want to get a population that is, you have confidence in, you understand that you're not putting something into the trial, i.e. a patient mix that isn't ideal to get the endpoint that you're looking for. But at the same time, you want it to be broadly applicable, because you know when you launch the product, you are just putting it out there to a broad audience. So if you make a narrow, if you really narrow your trial down to a very small group, then you either have narrowed your indication to your earlier point, i.e. choose your indication carefully, which may be fully appropriate for that asset, and you don't know unless you study it in a broader population. So you either segregate your population by a certain marker, i.e. a genetic marker, and say who has what genes, and therefore you're going to look at that as a personalized medicine approach to treatment. But you've got to have good data going into that to know whether or not those are the things you need to be looking at. So I think we are trending more toward looking at the patient population and defining a very careful patient population that will receive benefit from a therapy. Having good data going into that decision is not always so easy. And often, and certainly in the trials that we fail, we like to look back at and say, who in the population did succeed?
Yes.
And we learn a lot from that, but that's an expensive lesson. So we want to be able to develop the tools to be able to learn that as we go, rather than have to retrospectively look at it and say, okay, well, if we'd run the trial this way, we would have succeeded.
Can't you do that in that iterative manner where you're going back and forth, Kelly?
On the subgroup analysis, which I always would call then hypothesis generating. I think small companies, not large companies, small companies make the mistake of saying it's worked. And I'd say it hasn't worked because that was not the experiment. And I would say my first-hand experience has been about 50-50 running through the subgroup analysis, which is, not the analysis, running the new experiment, where these have just been false distributions that are not replicated in a subsequent trial. I think, again, I remind small companies these are hypothesis generating. They are not taken as, but companies raising money, trying to do partnerships, raise capital, convince clinicians to get involved, are representing them, I think, a bit more aggressively than they should otherwise be.
I also want to make a pitch, though, for the importance of prospective registries that examine the heterogeneity of patient populations and record data. These are extremely valuable, and you know, in my own field in spinal cord injury, we've seen that we've been able to really gain a much greater understanding of these different types of patient populations and the types of outcome measures that one can use, and there's a great value to looking at that, and I think that there needs to be greater investment in terms of collecting these types of data and doing the analysis, kind of really understanding the heterogeneity of that patient population. And I think in some regards we've tried to push too quickly along, trying to test a product in very broad patient populations, such as stroke, which is probably the prototypical example, without really fully understanding what the nature of that disorder is and where the subgroups are. And we've learned that bitter lesson, but I think it's important that we have the infrastructure in place to actually fund the collection of those data and the analysis of those data, to link those to the models of disease. And again, I'm just going to stress we need to do more smaller phase clinical trials, and those have to be designed better so that we actually get more information out of them beyond simply just saying, oh yes, it's safe and we don't have a problem and let's just forge ahead.
No, I think that's a really good point, Michael. And as you know, OBI and Terra Brain Institute is exactly using that as its model to try and establish that, where we can try and do both these kind of prospective, but also to end up doing both a data hypothesis and a data reverse engineering approach precisely along those lines.
Yeah, I would absolutely echo that from our point of view. The registries are huge opportunities to understand diseases better, and as we've talked about, there's many areas of neuroscience where we do not have that understanding today. And it's not the only area by any means. There's huge value in these things that it's a matter of having them at hand when you need them, because often we don't have them. And investing in them is something I've always been a champion of, but it's not about, you don't get a product out of the end of those investments. You have to see that's a long-term thing. So coming back to one of my early points about these public-private partnership concepts, and it's got to be, those are the kind of things that multi-companies and governments and to really understand disease get huge benefit from. And OBI, I agree, is taking the right approach to look at this from a broad point of view, collecting and holding data that can be mined and analyzed in ways that we just don't have the power to do today. And great dividends will come from that in the long run.
I just want to say that I agree with that, Todd. I just want to mention to people in the audience, they may not be aware of a true Canadian mega project that has really just launched called the Canadian Longitudinal Survey of Aging. This is a study of 50,000 Canadians. It's received $25 million in federal funds that are administered by CIHR but are outside of CIHR's budget. Basically, these 50,000 Canadians will be studied for the next 20 years. You enter the study at various ages from 45 to 80. You come back every 3 years. You get asked, well, literally thousands of data points are collected. How's your bone density, sir? How's your vision? How's your hearing? How's your cognition? How's your heart? How fast can you, walk 50 meters? And we're pretty excited about it. Blood is being drawn from these patients not only at the first visit, but at subsequent visit. So from the neuroscience point of view, half these people will develop stroke or Alzheimer's or Parkinson's disease in the next 20 years. So you can think of it as the beginnings of a registry. It's a prospective study in that sense. But it's an opportunity, again, to mine these data. So we're also going to be collecting genetic information, lots of other biomarker information from this population. So it's something that you can think of as you're thinking of ways from the startup perspective of providing better data for clinical trials, providing better endpoints. This may be a database that you can mine, and it will be collected with government funds.
So speaking of funds, I'm shifting to the next topic, money. Kelly, just tell us from your viewpoint as an entrepreneur, what's the environment like? What are the issues? How do we, this construct of partnership has come forward? So start us off in the discussion.
Sure. I mean, venture right now, I'd say broadly in North America and Europe, is going on under a massive restructuring as we speak. I mean, there are certainly areas of peak and interest. This week Facebook's going to get a price, and a lot of groups are going to be happy that they were part of Facebook. But I think a lot of other sectors, ICT, clean technology, and life sciences, find themselves. It's a different day and a different era in VC generally. I mean, the providers of capital for venture capital have typically been large pension funds, large banks, endowments, and the like. And those groups who would allocate maybe 0 to 5% of all the assets that they would have under management to venture capital are allocating a lot less to venture capital these days. For a variety of reasons. One is they don't like the volatility of venture capital, which is it is a boom-bust business. They don't like the lack of yield in venture capital, which is, what do you mean I've got to wait 8 years and figure out if my dollar that I invested today turns into $5 or turns into $0? And three, I think what's which in venture capital, what life sciences suffers from a little bit more than others, although I think it's we'll get to it in a second, but what it suffers from is there are not very traditional operating metrics for the progress of a life sciences company that an institutional manager at a big pension fund, be it the Canadian Pension Plan or be it CalPERS, can sort of monitor. It's someone saying, hey, we did well on a Petri dish, hey, it's looking nice in a mouse or a rat, hey, and progressing along. And that doesn't really, that may be scientific progress, and in fact it may be commercial progress, but to someone who's looking at revenue or margin or profitability or EBITDA 10 years hence, that's a tough story to communicate. So they tend to be, I think, in those places, which is we should have an allocation for venture, we should have an allocation for healthcare, and the healthcare value proposition is, sadly, there are a lot of diseases that need better treatments and or cures, and or we should be able to deliver healthcare at a lower cost than we currently do. And I think most healthcare opportunities that we all look or all have been involved in answer one, two, or three of those issues. Venture in Canada, so I started with a group in '96 called MDS Capital. I got to venture, I was fortunate to work in a lab of this gentleman, Peter Hislop, and he discovered a couple genes linked to Alzheimer's, and it piqued my commercial interest. And so I got into venture from there. And it was a different, in the mid-'90s, I'd say that was the beginning of the growth of venture. And I don't think we're at the tail end. I think we're just at the restructuring of venture where it's probably reached, I would say now, its set point. We are not going to turn back to the roaring days of '99 and 2000, or if we did, I think that'd be great news for everyone in this audience. I think, however, entrepreneurs and people who've been around for the last 5 years realize this set point where we are today is likely the set point for the next little while, which means a lot less risk capital going into these areas. And so what you see from that is fewer groups, but likely investing a little bit more money. You do see the emergence of pharmas and strategics coming in, and I wouldn't say replacing venture capital, but getting involved a lot earlier in the investment cycle than they currently have. And I think ultimately it's going to lead to a better rate of, I think the hope is it leads ultimately to a better rate of return, because I think the community will be at large, I'm talking the broad VC community, will be a lot more selective of the opportunities that do ultimately get funded.
Comments?
I can certainly comment on the pharma side of things, because I definitely myself have seen the trend where we're getting more active in venture, and other companies are getting active in venture. And it really, like any investor, I think comes down to the fact that we're no longer single-minded focused in how we invest in I guess what I like to term the healthcare ecosystem. We invest in research at the basic level. We invest in clinical trials. We invest in companies and partnerships when they're at a certain point. There was very little in the in-between stage, that startup company stage, for a long time. But as we look at the broad portfolio of what's going on, some companies are doing that, not linking it to their pipeline. Other companies are doing it, linking it directly to their pipeline, and saying venture is a space we need to be in to make sure the flow of knowledge, ideas, and innovation continues. And so it is, I would think, a positive trend in pharma recent.
Yeah, absolutely. I mean, I think we've had a couple companies. One got capital commitments from Medtronic, another from Lundbeck, Shire, and Takeda. My view is if you're going to get strategic investment, get more than one. One comes with typically some type of encumbrance, and you really don't want any encumbrance. So you invite more to the table. But I think it's an increasing trend that we're only going to see more and more of. And you do see that in MedImmune, Lilly, Pfizer, a lot of these groups, GSK, a lot more getting active. Maybe not at the start-off interface, but shortly thereafter.
Well, I think that's all. I think that's great, and I think it's very important that one have the opportunity to partner not only with providers of capital like venture capitalists or investment bankers, but also pharmaceutical partners, because they actually bring a huge amount to the table. They, our ability to screen compounds is 1/1,000,000th of your ability to screen compounds, and we should recognize that. We're probably better at finding the initial target, but after that, there's a lot of synergy. I just want to raise one other point, though. I mentioned my day job. I'm director of a brain research center. I just want to tell a story about some of our scientists who have basically come up with a whole series of exciting new targets and even lead compounds in several different indications. And you say, okay, and so we filed our patents like good boys, and we're working with something called the Center for Drug Research and Development, doing toxicology and distribution and therapeutic index, these kinds of things. And what's happening now, I'll just tell you what I'm struggling with personally. It looks as though the technology may well be commercialized in China. A lot of these guys are Chinese. They come back with max. We can get 100 million yuan in Guangzhou with no strings attached. We have to move. We have to spend time in Guangzhou. We've got to set up the R&D in Guangzhou. But the reality is that the 100 million yuan, even though it only translates to $16 million, probably goes as far as 50 or $60 million because everything's cheaper there. So what do? I'm the director of the center. The intellectual property will still reside in our university. That's the good news. But the wealth creation will be in China rather than North America. And I look at you gentlemen who are at some level the providers of capital. I guess I look at anybody from government who's in the audience. And I say, this is a challenge to us because I cannot in good conscience advise these leading scientists in my center just wait, Todd and Kelly will be over soon. I don't think that's going to happen. Go ahead.
I think one of the big challenges and opportunities that we have as Canadians. What's our position here as a kind of small to medium-sized power, and where are we? And I fully agree, because what was absent in the discussion was the emerging world. So I was just in Dubai, and Dubai runs on money, and certainly Asia and the BRIC countries is where the money is, where the investment is. But what they lack, and what we have here in Canada, is the intellectual capital, the intellectual firepower. And this is worth something. And I think we need to also be very realistic about what our position is and what the products are. So the intellectual ability and the knowledge and the clinical knowledge and the ability to run clinical trials, the ability to have these clinical cohorts is worth money, and I think we've underrepresented our own value in the world. So I think we need to start getting smart in this regard and start being realistic about how we can position ourselves. And certainly, I've seen this in the Middle East, where they're very aggressively pursuing these multi-specialty hospital systems, and they're very aggressively pursuing the expertise. They've got the money, but they don't have the intellectual ability to run these centers, and they don't have the clinicians, they don't have the scientists to do this. And this is potentially where our unique position might be. And I think we need to figure out how to effectively market that to the world, and how to actually effectively position ourselves along that strategy, because otherwise we're not going to be able to compete.
Our market is our knowledge.
Our market is our knowledge, and I think that is our product. To be quite realistic about that, because we're not going to outcompete Brazil or China or India in terms of making the widgets, but they're having difficulty competing with us on the intellectual level. So I think, how do we market ourselves effectively in that regard? And I think if we can figure that out, we will be effective. And I would argue that there's nothing wrong with getting the money from Guangzhou, but the trick will be, how do we try to bring that money back into Canada, and how do we harness the intellectual capital that we have here to leverage that? That's going to be the real trick.
And so our jobs may be different kinds of jobs, is also what you're saying. Todd?
First of all, I completely agree that we're seeing that trend. That trend is a real trend, not only if you look at it just from where someone might be going, but even within a large multinational company like my own and many of the others that we compete with. We internally, we look for how do we most effectively and economically do our clinical work, and we compete with internally we compete with other countries. Canada, as a clinical site, as a clinical organization, competes with other countries to have those patients in the trials. Now, no one country is going to win over any other in terms of having all the patients, but when it comes down to economics, it's really hard to compete when you're looking for high volume patients. In some studies with 10, 11, 12,000 patients, where are you going to spend all that money? Well, you could do it in North America and Europe, which is the traditional model, and that would cost you 3 times as much as if you went to these other countries. And that's a big internal problem because, again, we're constrained for resources. So it's not just about, it's actually within a company that same issue occurs. I do agree that there's got to be a balance, and it does land in the world of research. A phase 3 study where volume is a big opportunity, maybe that we don't have as good a competitive standing, but in a phase 2 world where there's a lot more science that goes on in that stage, we have a much better sense of, well, we bring more science to that discussion at that point in the discovery chain. So there's places to find your niche, find where your intellectual property, if you will, intellectual ability adds value, and where it doesn't, because we don't necessarily always want to be the place where everything gets done.
Time, and I'm also considering we had one more topic, which is the pipeline. And I don't know if, I actually think we've covered a fair amount of that, but I think what I'm also hearing is that in the pipeline, we have the potential of a unique niche that we have to take advantage of our skills, and that's early. Early and working together in a very collaborative way. So yes, you were going to
add to that. I mean, I think Health Canada has our regulator has been very helpful in that regard, in that a lot of our companies, US and Canadian for that matter, who would try, get in front of the FDA or wanted to, a lot of the first-in-man studies done accomplished here in Canada, where they will have holdups with the FDA. I mean, still today, I mean, the world is changing in front of us, but half the healthcare dollar is spent in the US. And more importantly, that's where the profit is made. And so when we look at our investments, we still have, that is shifting slowly, but that still is the large piece. So being very close to the US and the US centers is important for many of our companies. But curiously, a lot of the companies in Philadelphia, Boston, San Diego are getting first-in-man studies done up here in Ontario. Part of it's the strength of the clinicians and the scientific work up here, but mostly there have been a lot of delays within the agency that Canada has benefited for the moment.
So I'm now, I think I found this absolutely fascinating, and I don't know about the audience, but I'm going to open it up to comments and questions. If you do, you have to come up to the mic and please state your question there. You can identify yourself if you so wish. If you don't want to, you don't have to. But we do want you to speak in the mic because these are being audio taped. We would like to have everything on one document. So if anybody has comments, please, or questions for the panel or on their own. Don't be shy. No.
Waterston. I'm with the Center for Imaging Technology Commercialization, and I have a question about, we're often looking to the model south of the border where the SBIR program can play a very big role in terms of seed financing. And if you're looking at the Ontario environment, where do you see those types of opportunities looking most successfully for funding at that stage?
Where do I see them funding today or over time? I mean,
well, currently and in the near future.
I think it is the government's role to, in this realm, should be a provider of applied research grants. I think you do see the emergence, and I may not be spot on, so you may want to ask a follow-up question, or my panelists can fill in, but you do see bodies like HTX, FedDev, OBI, in fact, providing very targeted capital to projects that are applied for. It may not mimic the SBIR, but I think that is a role for government and government role funded agencies to be doing. And so I was, I can't remember how many years ago, TPC, Technology Partnerships Canada, decided to focus exclusively not only on aerospace, but also that meant Quebec. But that was unfortunate that those types of programs, I think we need, in a world where venture capital and partners are coming in, but they're coming in later than they would. The value that Max talked about of, hey, I've got an interesting target, I've got some leads, I've got some chemical matter. that's a world 10 years ago we would have funded. That's not a world we're funding anymore, and I think there's roles for those bodies to come in that slot right there.
Hi, I'm Jim Peters with the Baycrest Center for Brain Fitness. I don't think I ever heard the word software. I heard about mining and I heard about data a lot, but I'm wondering if you guys can make a comment about the role of software in software engineering in all of this, trying to capture all this data and figure out how to mine it and stuff.
I didn't mention the word software, but I was alluding to this whole point. I was kind of talking about analytical methods to deal with the heterogeneity of the data that are arising. And I was just at a seminar in California where the group at UCSF is using a principal components analysis to look at very heterogeneous outcomes in various neurotrauma models of various species and various outcomes. It does require, however, the sharing of information to drive that data. So that's a really critical point because the typical research articles will not have that. And so one of the points to drive all of this is really, I think, networks. And you need to have networks where investigators are willing to share data. And I think this is potentially one of the real roles of government to drive this. And industry isn't going to do this. And this is where we talk about the partnerships, but to be really practical, I think what this actually relates to is driving these types of research networks from an academic type of a basis, but where this information is shared. But I fully agree with you. And so what this group was showing in kind of just one aspect of kind of CNS disease was using really, fairly, I guess, in concept simple, but in practicality rather complex mathematical algorithms to come up with one number that would actually predict an outcome related to something as complex as traumatic brain injury.
I think there are, I think one of the great things about the brain is you can think about it as this, gooey organ full of genes and proteins and enzymes, but you can also think of it as a computing machine, and you can take advantage of that. So there are many ways in which you can use, have software lead to commercialization in neuroscience, I think. The second company that I founded was actually a hearing company. We developed algorithms that mimicked the functions of the A1 auditory cortex. It turned out to be very effective at identifying specific classes of sounds and locating them. So everything from, if you wanted a bullet detector, you could figure out where somebody was shooting at you from, or that was one indication. We developed something that could distinguish individual people from each other, and in the end the technology wound up not in neuroscience. We started it hoping to make a hearing aid. In the end it wound up in the car market, so you never know where it's going to go. And that's a lesson that you learn, I think, repeatedly. But there are many uses. If you can, one of the things I mentioned earlier, I didn't say software, I admit it, but I did say well-characterized cognitive test. That can be just as good as imaging if you can get, or better, as an endpoint. And there are many opportunities, I think, in that direction, from the posit science driving test and education, driving skill improvement technology to dozens of cognitive enhancement companies. It's got its own interesting set of issues, but certainly that's a great opportunity, and also the opportunity to develop other kinds of software-based tools which can be used in neuroscience and other areas.
Todd, did you have a...
I have one comment just around clinical trial design and experimentation. We're in a fairly conservative industry when it comes to the types of experiments we can do from an approval point of view. And that basically cascades all the way down through the different phases of development. There's lots of opportunity for using better tools to design better experiments to get better results. It's just the whole system has to move along in that direction. No one entity can do it.
We're going to take one last question, I think.
Thanks. Kelly mentioned at one point that if you're getting a strategic investment, you should try to get more than one. And I was wondering if the panel could talk a bit about that and what your thoughts are there.
Sure. Well, I'll kick it off, but then a single strategic investor will typically say, well, I'm interested not to just invest the capital, but I'd want access to something that you have. And that will come with what I'll loosely call encumbrance, which is, the worst would, the ultimate encumbrance is they would acquire the company and acquire the IP. But at some time, they just may want a place at the table. But the question is how great a setting you should provide them. Bringing multiple partners to the table, multiple strategics to the table. In the case of, so in one situation we're involved, this is where Shire, Lundbeck, and Takeda all have a place at the table. They get a place at the table, but they don't have any rights. And they all know that ultimately if these assets and IP are monetized, it's not going to be split amongst them. It's going to go to one of those groups. And so I think that's, in my mind, the idealized outcome. However, I think most of them don't like to do that. They wouldn't, we'll be the investor, and that it's on those conditions only. I will not co-invest with another of my peer groups. I think increasingly in this world of more cooperation in pharma, you're going to see more of the former, not the latter, but I think the latter still does exist. I mean, Todd, you'd be able to bring it a better perspective.
Absolutely support what you're saying. So if we're this single source of funding for something, then we expect that we are the recipient of the benefit, if you will, at the percentage or whatever is negotiated in. But as I was saying earlier, we're open in today's environment to look at some challenges and problems and solving them in ways that are pre-competitive. And if you're in a pre-competitive space, you're not looking for IP as much as you're maybe looking for some return and some advancement in science. But if we were the only company, then the nature of the business would say, well, we're basically the sole funder, so we should be looking at the first right of refusal and all of the other elements associated with that. So, and we don't shy away from those ideas of syndicated funding and multiple companies, and that's not a problem at all. But if you're on the receiving end of that and you want a single big partner, then no problem. But if you're looking for having a negotiating tool, if you will, you're better off to have multiple people at the table.
So you can see why they've been such an excellent panel and why they've been selected. I'm now going to ask Dr. Mario Thomas to come up and make the concluding remarks.
I'd like to thank all the members from this panel for some quite interesting insights into the challenges and opportunities of commercializing neuroscience discovery. I know you have taken time from your busy schedule all the way from BC, so thank you very much for joining us this afternoon. We are very grateful for your time. I'd like in particular to thank Don Stuss from OBI for moderating this panel in such a fantastic way. Thank you very much, Don. And once again, OCE is extremely happy to be partner with OBI in moving forward some of these early opportunities that will come out of OBI. So again, thank you very much. I have a small token of appreciation for your time. Lastly, I want to thank the Advanced Health team at OCE who has put together this panel, Jennifer Moles, Laura Wu, Bettina Klenker, and Shantanu Mittal. Thank you very much, guys, for putting this together.